Hello everyone, today we're going to talk about metabolism. When we think of metabolism, we think of two main reactions, anabolic reactions and catabolic reactions. Anabolic reactions are reactions that would synthesize large molecules from smaller ones. For example, proteins are made out of amino acids. So if we take amino acids and link them together to create something large, such as a protein, that would be considered an anabolic reaction. A catabolic reaction, on the other hand, this is a reaction where you break down larger molecules into smaller ones. So if you eat protein and you go ahead and you digest the protein and you break it down into its building blocks, which are the amino acids, that will be considered a catabolic reaction. Anabolic and catabolic reactions are reversible. Anabolic reactions, we usually go through a lot of anabolic reactions during a state known as the fed state or the absorptive state. And that's the period of time following a meal. So you've already had a meal, and so you already digested your food. So now this time you will be actually just you will be absorbing and storing those nutrients, and you will actually start building new larger molecules. For example, you just ate a meal, and so you just broke down some of the carbs that you just ate. You broke down some of the proteins that you've just eaten. What are you going to do with all of that glucose now in your body and all of these amino acids in your body? You are going to use them for energy, but whatever is going to be left over, you're going to actually start turning them into larger molecules. So you're going to start turning the excess glucose in your body into a larger molecule known as glycogen. You can take the excess amino acids in your body and make proteins. So therefore, we say that during this state, we are going to be going through a lot of anabolic reactions. Catabolic reactions are mostly done within a period known as the fasting state or the post-absorptive state. So when you are fasting, that's a time when you don't really have enough nutrients in your bloodstream because you are fasting. And so what you're going to do to go ahead and get some energy, your body is going to take those larger molecules, those reserves that you have inside of your body, and your body is going to start breaking it down um, so that you can get some energy. For example, if you've been fasting and you haven't eaten anything in a while, your body is going to take the stored glucose, which is glycogen, and it's actually going to start breaking it down so that you can get energy out of it. And so, therefore, we say that when we are fasting, we go through a lot of catabolic reactions. Our diet usually consists of fats, carbs, and proteins. When we go through a catabolic reaction, we break down those macromolecules into their smaller building blocks. So fats are going to be broken down into fatty acids and glycerol. Carbs are going to be broken down into sugars, usually glucose. Proteins are going to be broken down into amino acids. Now, the plasma amount of glucose, so our plasma glucose concentration, is the most closely regulated one because glucose, turns out that glucose is the only fuel your brain can metabolize. Except in times of starvation, your brain can actually also use something else known as ketone bodies. Otherwise, your brain hugely relies on glucose. And so therefore, if this image right here represents the amount of glucose that we should normally have in our blood plasma, and all of a sudden your blood glucose drops way below that normal level, who's going to have access to that glucose? Only the brain. Now, if we have normal levels of glucose, then your brain is going to have access to it, but also all of your other tissues, they're going to also have access to that glucose. What if we have excessive amounts of glucose in our bloodstream? What's going to happen to all of that glucose? If you have excessive amounts of glucose, then it can actually be excreted in your urine. Or if you have excessive amount of glucose, you can actually start storing it as fat. And that process would be known as lipogenesis. Lipo coming from the word lipids, which are fats, genesis meaning to make. So excess glucose can turn into fat and get stored in your body as fat. What else can happen to excess amount of glucose? We can go ahead and store it in the form of glycogen. The process of converting the glucose into glycogen is known as glycogenesis. Again, genesis meaning to make, and so we are making glycogen. Glycogen is usually stored in your liver and in your skeletal muscles. Okay, so we've already talked about what can happen if your glucose levels are in excessive amounts. Well, let's go ahead and talk about what could happen if, let's say, you are fasting. So if you're fasting, your glucose levels in your bloodstream are actually going to be pretty low. So if your glucose levels in your bloodstream are pretty low, what is your body going to do? Well, your body is actually going to go and access the glycogen, pull out the glycogen out of your liver and out of your skeletal muscles, and your body is actually going to start breaking down the glycogen back into glucose so that you can start increasing the glucose levels in your bloodstream. That process of breaking down the glycogen back into glucose is known as glycogenolysis. If we break the word down, glycogen is right here, and then the suffix of the word is lysis, which means break down. So glycogenolysis is when we break down the glycogen into glucose. Now, also, if your blood glucose levels, it's pretty low, other than going through glycogenolysis, we can actually go through something else. As we said, proteins are made out of amino acids. We can actually take the amino acids and convert it into glucose. And that process is known as gluconeogenesis. Let's break the word down. Gluco meaning glucose, neo meaning new, genesis meaning to make. So we are making new glucose. And we use that term gluconeogenesis usually when we are making glucose out of a non-carbohydrate source. So in this case, if we are using the amino acids to make glucose, that's a non-carbohydrate source right there. And so that process is known as gluconeogenesis. Amino acids, usually, if we have amino acids, usually they are going to make proteins. So as you can see, we can recycle our amino acids and make proteins. But in case, as I said, you have very low levels of glucose, then amino acids can go ahead and convert into glucose via a reaction that they will go through. If we take a look at this image right here, we see that fats are also going to be broken down and used by your tissues. So your tissues can also use fats and break them down and get a lot of energy out of them. Glycogenolysis is the process of breaking down glycogen. So if our glucose levels in our bloodstream is pretty low, we go through glycogenolysis so that we can get the glucose, which is then going to be used for us to make ATP, which is energy. Now, when glycogen is broken down, glycogen can break down into plain glucose, and that's 10% of the time, or 90% of the time when glycogen breaks down, we get what's known as glucose 6-phosphate. Now, glucose 6-phosphate is what's going to be used during our cellular respiration to make energy. And so therefore, if glycogen is broken down into plain glucose, we must convert that glucose into glucose 6-phosphate by adding a phosphate to that plain glucose. Breaking down glycogen into glucose 6-phosphate, it's going to be faster for us to use it directly to make energy, versus if we break down glycogen into plain glucose and then we have to convert that glucose into glucose 6-phosphate, it's going to take longer, and it's going to take more energy from your cell to actually do that. But at the end of the day, after we get that glucose 6-phosphate, either directly or by extra steps, that glucose 6-phosphate will enter into the glycolysis process during cellular respiration, and we go ahead and we make lots of ATP. As we said, proteins are made out of amino acids. So when you eat protein, you break down the protein into amino acids. Those amino acids can further break down, so they can go through more catabolic reaction and break down further into ammonia and a bunch of organic acids. And that process is known as deamination. Now, those organic acids that would result can actually be used by your cellular respiration reaction, so we can use them to make energy. And also, if your glucose levels in your bloodstream is pretty low, then we can use those amino acids after we break them down and they will go through more reactions. We can use them to convert them into glucose. Also, if you have, let's say, lots and lots of amino acids in your bloodstream, what can we do with all of that excessive amount of amino acids? They can actually be turned into fat. So you have to kind of be aware of how much protein you are consuming, because if you are consuming way too much protein than your body really needs, then all of that excessive amino acids from the protein can actually be turned into fat. That ammonia, by the way, that would result is toxic, so your body converts it into urea, and eventually your body will excrete it out. Let's say that you have an enzyme number 1 right here, and this enzyme, its job is to speed up the reaction of converting substrate A, into substrate B. But let's say you also have another enzyme number 2, that would speed up the reaction of converting B back into A. Now, if you have equal amounts of those enzymes being released at all times, you're never going to have a net amount of A, and you're never going to have a net amount of B, because you are always going to be going through that cycle back and forth, converting A into B, and right away converting it back into A. So if you want to control how much substrate A or how much substrate B you want to have, you really need to control how much of those enzymes you are releasing. So let's take a look at an example. Let's say that we are in a fed state, or basically absorptive state. That's when we are in a state where we have just eaten a meal. Now, if you've just eaten a meal, what's going to happen to your glucose levels? Well, your glucose levels are going to start to increase, okay, after eating a meal. Now, high glucose levels in your bloodstream, that's going to stimulate your pancreatic cells to produce a hormone known as insulin. Insulin is going to start decreasing your glucose levels in your bloodstream. How's insulin going to do that? Well, insulin is going to stimulate a bunch of enzymes, and those enzymes are going to take the glucose in your bloodstream and start converting it and storing it in your liver and in your skeletal muscles in the form of glycogen. So insulin is going to increase the levels of glycogen in your bloodstream because it's going to be promoting the conversion of glucose into glycogen. Now, also, insulin is going to inhibit the enzymes that will break down glycogen into glucose because you don't want to increase your glucose levels. The whole point of producing insulin was to decrease your glucose levels. And so insulin is going to prevent the enzymes that will break down glycogen back into glucose. Let's say that right now you are fasting. If you are fasting, what does that mean? It means that your glucose levels are going to start to drop. If your glucose levels start to drop, your body would want to increase your glucose levels. And so your body is not going to produce insulin. Low glucose levels are going to stimulate your pancreas to produce another hormone. That hormone is known as glucagon. Glucagon is going to do the exact opposite of insulin. Glucagon is actually going to access the glycogen that you have stored in your liver and in your skeletal muscles, and it's going to stimulate a bunch of enzymes that will break down the glycogen into glucose so that you can increase the glucose levels in your bloodstream. And glucagon is also going to inhibit the enzymes that will try to convert the glucose back into glycogen. Because right now, your body wants to increase the glucose and not really store it. And so therefore, when glucagon is released, your net glucose synthesis is going to increase because glycogen is going to start breaking down into glucose. When insulin, on the other hand, is being released, your net amount of glycogen is going to increase because your body would want to store the excess glucose as glycogen. So as we said, your pancreas has the ability to secrete and release two different hormones, insulin and glucagon. Insulin is going to be produced by cells in your pancreas known as beta cells, and glucagon is going to be released by cells in your pancreas known as alpha cells. And we know now that when is insulin going to dominate? Insulin is going to dominate after you've eaten a meal, right? So when you are in the fed state. Glucagon is going to be dominated when you are in the fasting state. So when you're fasting and your glucose levels drop, glucagon is going to be the dominant hormone being released so that it can help you increase the glucose levels. And so therefore, during the fasting state, glucagon is going to dominate and insulin is not going to be released, and vice versa. After you eat, insulin is going to be produced and glucagon is going to be inhibited. Okay, let's say that you are in a fed state. Basically, you've just eaten. If you've just eaten a meal, then we know that your glucose levels are going to increase. And if your glucose levels increase, then the hormone that's going to dominate is insulin. Insulin is going to try to decrease your glucose levels in your bloodstream. So if we take a look at this graph right here, and this dotted line is showing you your glucose concentration after eating a meal. You notice that after you eat a meal, your glucose concentration increases. And so if your glucose levels after eating a meal increases, the hormone that's going to increase in production is going to be insulin, because insulin would want to try to lower that glucose level in your bloodstream. The hormone after a meal that's going to be pretty low is glucagon, because glucagon, what it does is increase your glucose levels. And right now, your glucose levels are already high, so you don't want to produce glucagon. So after a meal, insulin is going to be pretty high, and glucagon is going to be pretty low in your bloodstream. Now, what insulin does, as we said, it's going to try to decrease your glucose levels. It's going to try to let your cells absorb the glucose, and we will talk about how is that going to be done. And it's going to try to increase your glycogen synthesis. How it's going to try to, as I said, stimulate a bunch of enzymes, and those enzymes are going to convert the excessive glucose now that we have in our bloodstream into glycogen, and that glycogen is going to be stored in our liver and in our skeletal muscles. If you have too much glucose in your bloodstream, insulin is going to increase the fat synthesis process. And so, therefore, we have to be careful about how many times we're eating or how much we are eating, because if someone constantly eats, even though, let's say, they are eating healthy things, but you are still constantly eating and your glucose levels keep spiking up, then insulin is going to keep spiking up, and then all of that glucose is actually going to start getting stored in the form of fat. Insulin also increases our protein synthesis process. Now, on the other hand, if we are fasting and our glucose levels drop, so if your glucose levels drop, you definitely don't want to produce insulin, because insulin is just going to make things even worse. And so your pancreas, during fasting, your pancreas is going to produce the hormone known as glucagon. Glucagon is going to do the opposite. Glucagon is going to go to the stored glycogen in your liver and in your skeletal muscles, and it's going to break it down, the process known as glycogenolysis, as we said, and it's going to break it down into glucose to increase the glucose levels in your bloodstream. Glucagon is also going to increase the process known as gluconeogenesis. If we remember, gluconeogenesis is the process of making glucose out of non-carbohydrate sources. So if your glucose levels in your bloodstream, their levels are really, really low, your body will go ahead and take some of the amino acids you have and convert those amino acids into glucose. Another thing your body is going to do to try to increase your glucose levels is that glucagon is going to help the process of making ketone bodies. How do we make ketone bodies? Out of fatty acids. So fatty acids you get fatty acids by breaking down fat. So your body is going to break down the fat for you. You get fatty acids out of it. You take those fatty acids and you convert them into glucose. And you don't convert them into glucose, you convert them into something known as ketone bodies. And those ketone bodies are going to be used by your brain. So as we said, your brain highly depends on glucose as its major fuel source. So if your glucose levels are really low, as we said, your body can either take the amino acids, convert them into glucose, But if that's not enough, your body is going to take the fatty acids and convert those into ketone bodies. And then your brain is going to be able to use those ketone bodies as a fuel source. Now, there are diets out there that limit your carbohydrate consumption, and you depend highly on eating fats. Now, this could be dangerous because too many ketone bodies in your bloodstream, because now you're eating too much fat and you're breaking that fat into fatty acids, which will convert into ketone bodies. Those ketone bodies can start making your blood highly acidic, which is dangerous, and that would be known as ketoacidosis state. Some cells in our body, such as our adipose cells and our skeletal muscles, especially when they are in a relaxed mode or resting mode, they need insulin to help them absorb glucose. Without insulin, those cells are not going to be able to absorb the glucose out of our blood plasma. If we look at this image right here, when we are in a fasting state, we know that when we are fasting, our glucose levels are pretty low. And so if our glucose levels are pretty low, then insulin is not going to be released. And so if insulin is not going to be released, no way would those glucose molecules be able to enter into the cell. And so they will stay outside of the cell. Now what happens is when we go ahead and we eat, our glucose levels are going to increase. But for those glucose molecules to actually enter into the cell, insulin is required. So when your glucose levels increase in your blood plasma, insulin is going to be released by the pancreas. Once insulin is released by the pancreas, it's going to go ahead and bind to a specific receptor on the cell surface. Once it binds to that specific receptor, a bunch of cascade events will go ahead and take place, causing those vesicles inside of the cell carrying a bunch of glucose transporters known as the GLUT4 transporters. Those vesicles are going to get close to the plasma membrane, and via the process of exocytosis, those vesicles are going to release those GLUT4 transporters on the surface of the cell. Once those GLUT4 transporters are inserted on the surface of the cell, glucose is going to pass through those GLUT transporters into the cell. And therefore, the glucose will only be allowed to enter into the cell only after insulin binds to that specific receptor on the cell surface. Other cells don't need insulin for glucose to enter into the cell. And we're going to go ahead and take a look at an example. Liver cells have GLUT2 transporters that are found always on their cell surface. When we are fasting and our glucose levels are pretty low, we are definitely not going to be producing insulin. Instead, we are going to be producing glucagon, and glucagon is going to help convert the glycogen storage that we have into glucose. And so what's going to happen is that the level of glucose inside of your liver cells are going to be much higher compared to the level of glucose outside. And so those glucose molecules are actually going to make their way out through the GLUT2 transporters out of the liver cell so that they can transport the glucose outside of the liver cell to try to increase the glucose levels in your blood plasma. Now, after we eat a meal and our glucose levels increase, that high level of glucose is going to stimulate the production of insulin. Once insulin is released, it's going to bind to receptors found on the liver cells, causing the glucose to actually start entering into the liver cell via the GLUT2 transporter. And once the glucose enters into the cell, it's going to be converted into the glucose 6-phosphate, which is going to be used up in our aerobic cellular respiration process. Um, and we're going to start making ATP. And actually, by converting the glucose into glucose 6-phosphate, that's always going to keep the free glucose concentration low inside of our cell compared to outside. So more glucose keeps entering into the cell. Glucagon is produced by our alpha cells within our pancreas. What stimulates the production of glucagon is low plasma glucose levels, because we know that glucagon tries to increase the glucose levels in our blood plasma. So low levels of plasma glucose can stimulate the production of glucagon. Another thing that can stimulate the production of glucagon is when you have too much plasma amino acids. Once glucagon is produced, we know that glucagon is going to go to the glycogen storage in your liver and in your skeletal muscles, and it's going to try to break down the glycogen into glucose. And so glucagon is going to definitely increase the process of glycogenolysis, the process of breaking down glycogen into glucose to try to increase your glucose levels. Another thing is that when your plasma glucose levels are pretty low, your insulin, your beta cells within your pancreas, they're going to be inhibited from producing insulin. And so you're not going to be producing insulin if your blood glucose levels are pretty low. And so what's going to happen is that your body, to try to make up for that lost or for that low levels of glucose, your body can also take the amino acids and the fatty acids in your body and try to convert the amino acids into glucose via the process known as gluconeogenesis. And your body is going to take the fatty acids and convert them into ketone bodies so that your brain can go ahead and use those ketone bodies. And definitely if you are in a state of hypoglycemia, hypo meaning low, glycemia meaning sugar. So if your sugar level is pretty low, then your brain is going to rely on the ketone bodies. Also, right here, this image shows a negative feedback loop. So once your glucose levels increase, then you're going to send a negative feedback signal to stop your alpha cells from producing any more glucagon.